Hello students. Today we're going to start BSc third year semester 6 Zoology Unit 10 Carp Hatcheries, Transportation of Breeders and Seed. Now this unit we are going to learn about the types of hatcheries, etc. So to introduce the topic, what are carp hatcheries? Carp hatcheries are specialized facilities designed for the controlled breeding, incubation and hatching of carp. These hatcheries play a crucial role in aquaculture by ensuring a steady supply of healthy fish for commercial farming, conservation, and research purposes. Now, let us look at the types of hatcheries: traditional hatcheries, Chinese hatcheries, and Indian hatcheries. Traditional hatcheries are nothing but natural ponds for breeding, whereas Chinese hatcheries, they are circular hatchery systems for controlled spawning. If you look into Indian hatcheries, they have flow-through systems with bund breeding techniques. What are the key functions of carp hatcheries? Brood stock selection. That means to say choosing healthy adult fish for breeding. Then the other function is induced breeding, using hormones or environmental triggers to stimulate spawning. Then is egg incubation, providing optimal conditions for eggs to develop and hatch. Then last one, larval rearing. So feeding and nurturing young fish until they are ready for transportation or stocking. Then what is the need of these hatcheries? Why do we need to set up these hatcheries? So in earlier days, availability of fish seed was entirely dependent upon wild catch from rivers and large reservoirs during rainy season, which was accomplished by several problems. And in present time, the quantity of seed is collected from rivery source and which is very low. Then fish farmer had to depend only on natural water bodies for their seed requirements. Then seed they collected constituted mixed variety of different species. Due to lack of knowledge, they reared mixed variety, which resulted in less survival rate. During the transportation of seed, there occurred high mortality of seed also. So, thus, for these reasons, hatchery construction is so necessary to overcome such problems and to obtain maximum quantity of seed in proper condition. Now, what is the special requirements to maintain proper hatcheries? Carp hatcheries require optimal environmental conditions to ensure successful breeding, healthy larval development, and high survival rates. Here are the key environmental factors that influence carp hatcheries. The one is water quality pH level. It should be, the ideal range is from 7 to 8.5 to support fertilization and larval growth. Then dissolved oxygen. It must be maintained above 4 to 6 mg per liter for proper egg incubation and fry survival. Then ammonia and nitrate levels. It should be kept low to avoid toxic buildup, which affects fish health. Then temperature optimal range also. It should be 24 to 30 degrees centigrade for induced breeding and egg development. Last one, temperature fluctuations. So sudden changes can cause stress or mortality in eggs and larvae. Now, if you see the water flow and aeration, it should be gentle flow. It ensures proper circulation in incubation tanks and prevents egg clustering. Then aeration also. Aeration system, that means to say we need to maintain oxygen levels using diffusers or aerators in hatchery ponds. Then light and photoperiod natural light exposure. So that helps in regulate breeding cycles. Controlled lighting should be there. Then artificial lighting may be used to influence spawning behavior also. Then next, salinity and hardness, and then freshwater conditions. Most carp species thrive in freshwater environments. Then water hardness. The hardness of water should be, the ideal range is 50 to 200 mg per liter CaCO3, essential for egg development. Then nutrition and feeding brood stock. High protein diet, that is 30 to 40% with vitamins, enhances egg quality. Larval feeding also. Live feed like zooplankton and formulated diets improve survival rates. Hatchery infrastructure and well maintained tanks and ponds also. So they prevent contamination and disease outbreaks. Next, proper waste management. It keeps the hatchery environment clean and stable. Maintaining these factors helps achieve successful fertilization, healthy larval growth, and efficient seed production for aquaculture. Now, what is essential for hatchery requirements? RAS technology. It's nothing but a recirculating aquaculture system. It is ideal for controlling the water parameters. It gives the young fish the optimal living conditions, resulting in healthy fingerlings. Then good genetics. Using broodstock from a well-performing genetic strain improves the growth and healthy performance of the fish. Then we also need to look into high quality fish feed. The broodstock and young fish in your hatchery should be fed with high quality fish feed to give them a head start in life. Quality fish feed is not only important for fish but also for the operation of the RAS systems, as low quality fish feed spoils the filter of the RAS system. Proper management and staff. A successful hatchery cannot work properly without good management and the well-trained staff. Now let us discuss about the types of hatcheries. The number one is earthen hatching pits. These are traditional fish breeding structures used for natural incubation of fish eggs, especially for species like carp. These pits are commonly found in rural aquaculture setups and are designed to mimic natural spawning conditions. Characteristics of earthen hatching pits. First, material. Material should be made from soil or clay, allowing water retention and slow seepage. Then size and shape. Usually circular or rectangular pits with a depth of 0.5 to 1.5 meters. Water source. It should be filled with fresh water from nearby ponds, rivers, or groundwater sources. Aeration, natural exchange of gases, occurs through soil absorption and surface aeration. Now, what should be the dimensions? The dimension for earthen hatching pit should be 3 into 2 into 1 inch or 8 into 4 into 2 inches. Based on the requirements, the size may vary. These pits are prepared in several rows and their inner walls are plastered with mud. Several pits are interconnected so as to allow the water to flow from one pit to another. In each pit, 3 layers of cloth tanks or harpas of different dimensions are tied. The outermost cloth tank is about 6 feet into 3 feet into 1.5 feet in size and is made up of cheap cloth. For support, 2 bamboo poles are fixed to which these cloth tanks are tied. The middle tank is smaller, 5 into 2.5 into 1 feet in size and made up of mosquito cotton cloth with round meshes. The innermost tank is 4 into 2 into 3 by 4 foot in size, having bigger mesh size. Then coming to hatching of eggs. After filling with water, the collected eggs are introduced. The eggs collected from rivers contain not only the eggs of major carps, but a mixture of eggs of several types of fishes and also certain larvae of insects, crustaceans, etc. The eggs thus, before transferring to the hatching pits, are subjected to sieving. A sufficient number of eggs, approximately 9 lakhs to 22 lakhs, should be kept in each hatching pit. Out of these, only 2.5 to 25% successfully hatch. The flowing water due to the interconnection between the hatching pits activate the eggs to hatch. The time taken for hatching ranges from 2 to 15 hours. Now, what are the advantages of earthen hatching pits? These are best suited for hatching eggs from dry bunds. Wide areas near dry bunds can be used for digging earthen pits so as to use a less quantity of eggs in each pit. Fresh accumulated rainwater from the bunds enters into the pits for hatching, and they are cost effective because it requires minimal investment compared to artificial hatchery tanks. Then they are also eco-friendly because they are made from natural materials, reducing environmental impact and temperature control also. Clay structure helps maintain optimal conditions for hatching. Higher survival rates. They provide a stress-free environment for larvae. Now, what are the disadvantages, or we can say what are the challenges and limitations? Water quality management needs frequent monitoring of oxygen and pH levels. Then predation and contamination, because open pits are vulnerable to predators like insects, birds, and frogs. Then limited control. Unlike artificial hatcheries, these pits have less control over environmental factors. Then how do they work? First, we need to, if you look at this, first we have to select the site, then we need to prepare the pit, then watering should be done, then eggs should be introduced, then finally monitoring and maintenance like water conditions are checked periodically to ensure high hatch rates. Now, let us see earthen pot hatcheries. Here, the principle of hatchery system is followed, where an overhead tank in the form of an earthen clay pot is installed on a brick stand about 2 feet in height. There are two different size containers. Container number 1 acts as incubator chamber, while container number 2 acts as a spawnery. From the bottom of the earthen pot, a valve and a rubber pipe are fitted. The other end of the rubber pipe contains a metal tube, which is put into container number 1. At the top of the container, a round mesh sheet is placed and it is held in position by a bamboo rod. At the other end of the container 1, near to the surface, a round spout is fitted. This spout from container 1 opens into container 2, which is smaller in size than the first one. On the top of the container 2, a net is placed and to its other end nearer to its surface, an overflowing pipe is fitted. The outer end of the pipe is covered with a small net. How does it work? So the working method is, at first fresh water from a tube well is poured into the earthen pot. The valve is regulated to maintain the water pressure entering into container 1. Fertilized eggs are then introduced into the container 1. The excess water flows out through the spout into the container 2 and from it to the outside through the overflowing pipe. A running water condition is thus maintained. The water pressure is regulated in such a way that the eggs do not pass out into the second container. When the eggs hatch, the hatchlings have a tendency to undergo vertical migration. So it comes to the surface and the overflowing water carries it into container 2. The unwanted materials such as eggshells, unfertilized eggs, deformed eggs remain behind in the first container. The spawns are then kept in container 2 for 2 to 3 days. It is fed externally if necessary and later are either introduced into the nursery ponds or sold to the other farmers. Now, what are the merits of this earthen pot hatchery? Cool and running water condition, which is essential for hatching, is maintained. Cost of making an earthen pot hatchery is very less. Very easy process to handle. Coming to demerits, Earthen pot is not capable of storing a large amount of eggs and very laborious manual process and time consuming. Next one, if you see glass jar hatchery. A glass jar hatchery is a much modified setup than the earthen pot hatchery. It is comprised of water supply system, breeding tanks, incubation and hatching jars, and spawnery. Glass jar hatchery is generally installed by the side of a water source, which may be a perennial pond or a river or a water supply from a tube well. It should be kept in a well ventilated room with adequate light and proper drainage system. Now, to how to construct this. First, overhead tank. So, available water from the source is pumped up into an overhead tank, which is installed above a brick wall. If water is drawn from a river, then a distillating tank should be provided. A fine meshed wire netting is provided in the pump system. This helps to prevent the entry of organisms commonly present in the water of river and pond. A deep tube well is of much facility as it provides clean water. The tube well should be installed in an arsenic-free area. The next one is breeding tank. As hatchery is a self-contaminated unit, it generally contains a breeding tank with arrangement for overhead showers. This tank is provided for spawning the fish. An outlet is present in each tank for draining the excess water. A breeding tank is made of 1.8 into 0.9 into 0.9 meters in dimensions. Generally, in a farm, there are 2 to 3 breeding tanks, whereas 2 to 4 breeding hapas can be fixed at a time. However, the number of breeding tanks in a hatchery depends on the spawn production target. In a hatchery where there is no provision for breeding tanks, the operation is undertaken in hapas fixed in ponds, and the fertilized eggs are released into the glass jar for incubation and hatching. Sometimes the spawnery itself can be used as a breeding tank. In a hatchery where there is no provision of breeding tanks, the operation is undertaken in hapas fixed in ponds, and the fertilized eggs are released into the glass jar for incubation and hatching. Sometimes the spawnery itself can be used as a breeding tank. Now, there are different types of incubation and hatching jars used, ranging from the glass jars, synthetic jars, to zong jars with temperature control with water supplies. Now, glass jars are conical in shapes. They are open at the top and gradually tapering towards the bottom. The glass jars are set up vertically through the circular holes made in a wooden table. The table is 4.05 into 0.6 meters with a height of 0.9 meters. The jars are fitted in 2 rows at a gap of 22.9 centimeters. They are kept in a vertical position with the help of clamps. The top of the jar is fitted with a galvanized iron ring with a beak. It serves as an outlet for water coming out of the jar. The jars are connected with a rubber tube at the bottom with respective tabs. Now, each glass jar has a capacity of 6.35 liters and can accommodate 50,000 carp eggs at a time. A long open galvanized conduit is fitted in such a way that the overflowing water from each jars passes into it and flows down to the spawnery. Next is jug jars. The jug jars, named after jug, where they were first used, are large, that is 60 to 70 centimeters in height and 15 to 20 centimeters in diameter. Inverted bottles with open tops and narrow bottoms. In a hatchery, they are installed vertically in a series with the narrow neck directed downwards, attached to the water lines with suitable taps. Water is supplied from below and the outlet is at the top. The jug jars have a capacity of 6 to 8 liters and can carry about 1 to 5 liters of eggs. Then the next one is McDonald or Chase jars. McDonald jars are similar to the jug jars. It is cylindrical in shape, that is 40 to 50 centimeter height, with a circular bottom for the water supply, and the flow of water in the jar keeps the eggs rotating slowly. Farm fabricated incubation funnels. They are made of plexiglass or fiberglass in any required dimensions and are widely used. Israeli farmers use flexiglass incubation funnels of 80 centimeter long and 60 centimeter in diameter at the upper rim. Water is supplied from a bottom inlet while water overflows from an outlet at the top. This overflowing water is used for collecting the hatchlings. Each such funnel can be used to hatch about 1,75,000 carp eggs. Oinorovic and Horath illustrated a number of designs of incubation funnels that can easily be made with plastic and sieve cloth materials, with some sprinkler type water inlet. Next is plastic jars. These jars are made of low density polythene. The upper part has a diameter of 17 inches and 24 inches height with a capacity of 40 liters. Each jar has independent control valve. For effective use, an inner egg container is used for separating the eggshells after hatching. Hatching jar with water supply from above is also in use when the inlet pipe from above extends deep into the bottle. Next, spawnery. A spawnery is a harp of synthetic material which is fitted on a frame placed inside a cemented cistern or tank. The height of the harpa is slightly more than that of the cemented cistern to prevent the hatchlings from being carried away along with the overflowing water from the cistern. An overhead shower is provided to spray water over the developing hatchlings in the harpa. The cemented cistern of the spannery measures 1.8 meter into 0.9 meter into 0.9 and the harpa measures 1.65 into 0.8 into 1 meter. It can accommodate about 10 lakh spawns at a time. Now, how does this work? Water either from adjacent ponds or rivers or from groundwater flows into the incubation jars and spawnery. The water hardened eggs, about 50,000, are then slowly released into each incubation jar and the water pressure is regulated during incubation so that the eggs are stirred gently without being split or overflowing. A flow of 600 to 800 ml per minute is sufficient to keep the carp eggs in circulation inside the jar. Water enters into the incubation jars either from the bottom or from above through a pipe which opens at the bottom. After the eggs are hatched, the hatchlings have a tendency of vertical migration. As a result, they come up to the surface and pass through the outlet along with the overflowing water into the open conduct leading to the spawnery. The rate of flow of water in the jars at this time is slightly increased after the completion of hatching to facilitate speedy escape of hatchlings. Dead eggs, unfertilized eggs, eggshells, abnormal and dead hatchlings are left behind in the jars, which are subsequently removed by disconnecting the glass jar or incubation jar from the tap. In the spawnery, the water is sprayed continuously for about 3 days until the fry are ready for stocking. Now, let us see the merits. It can be operated in a compact area. As the incubation jars are transparent, the developing embryos can be seen with the naked eye and appropriate rectifications can be made. If you look into the demerits, in case of incubation jars made of glass is liable to damage and cannot be shifted to different places. Temperature control system is not provided. Additional air circulation is not provided. Hatching success is less than that of Chinese hatchery. Damage of eggs from the glass jars during transfer may take place. Next, coming to cement hatching pits. The hatching pits are lined with cement. The eggs are kept in these pits for hatching. The main advantage of these pits are the recurring expenses are less. They are easy to operate and regular flow of water is maintained. But capital investment is high and the mortality is mainly due to depletion of oxygen and in water temperature. The percentage of hatching is 30 to 50 percentage. Now, next, let us look into floating harpas. Floating harpas are an improvement over the conventional harpas. These are designed to cope with the rise and fall in the water level. These can be easily fixed even in rocky areas without bamboo poles. They can also be fixed in deeper areas so that mild water current passes through the harpa. This helps in better exchange of water and aeration. It is similar to a conventional harpa, but it is mounted on frames which are made up of polythene or aluminum pipes. Floats are fixed to the harpa for floating. It is tied to fixed objects with long ropes so that it will not be carried away by the current. It is collapsible and can be assembled very easily. The size of outer harpa is 2 into 1 into 1 meter and that of the inner one is 1.75 into 0.75 into 0.5 meter. The hatching percentage is 50 to 70 percentage. Silt may get deposited in the harpa which causes mortality of the sperm. It may be dispositioned due to the movement of water and rearranging is time consuming. The hatching rate is not high. Next, coming to tub hatchery. This hatchery was introduced in Madhya Pradesh. It is an improvement over fixed harpas and provides for hatching in running water. It has a continuous flow of water by gravity and siphons. This system has a series of 8 to 12 galvanized iron hatching tubs connected to each other with a regular flow of water. Each series consists of an overhead drum. Each tub is 2.5 into 2.5 into 1.5 inches in dimension and has two nets, an outer and inner one. The fertilized eggs are transferred into the tubs for hatching. The percentage of hatching is 50 to 70%. Vigilance round the clock is necessary in this system. Next, plastic bin hatchery. This unit consists of 8 hatcheries cum spawnery units and a 5,000 liter water tank. The tank receives water from a natural resource by pumping. The tank is connected to the inlet pipelines of each unit. The HCS units can be arranged in a series to facilitate inlet connections. In this hatchery, 2 crore eggs are kept for hatching. The percentage of hatching is 70 to 80 percentage. Each unit consists of an outer container and the inner common egg vessel. The outer hatchery container is a rectangular aluminum sheet tub of 54 inches into 18 inches into 22 inches dimensions and 243 liters capacity. It is unequally divided into three chambers. At a time, 8 liters of eggs are placed for hatching in each hatchery unit. It also consists of an inlet, outlet, and drain pipe. Plastic bucket hatchery. It consists of an outer plastic bucket with a perforated aluminum bin egg vessel and a galvanized iron sheet spawnery. The plastic bucket height is 47 centimeters, 30 centimeters diameter, and the capacity is 45 liters. It has three inlets at the bottom and two outlets at the top. The eggs are kept in the egg vessel for hatching. The survival rate is 70 to 80 percentage. Next is galvanized iron jar hatchery. This hatchery resembles Baumik's glass jar hatchery, except that the hatchery glass jars are replaced by galvanized iron jars. Each jar has a cylindrical portion that is 48.5 centimeters long and 23 centimeters diameter, and a top conical portion that is 19 centimeters long. The jars are filled on an angle iron framework, which also accommodates a galvanized iron conduct, 10 centimeters wide and 10 centimeters height, to carry the hatchlings from the jars to the spawnery. The water flow rate is 1 liter per minute, and the galvanized iron jar hatchery is cheaper than glass jar or polythene jar hatchery. Next is Shirgur's bin hatchery. This hatchery consists of a rectangular aluminum container provided with circulating water in which they are placed cylindrical egg vessels. Each egg vessel can hold about 2 lakh carp eggs at a time. Next one is Chinese hatchery. The Chinese spawning and hatching systems are based on continuous flow of water by gravity to breed calves and hatch eggs. The cost of construction and operation of a Chinese hatchery is less when compared to any other design for the same production capacity. In India also, the Chinese hatchery system is now considered to be highly suitable for the production of quality fish seed. Chinese type of hatchery consists of four main components. Examples: overhead water storage tank, the spawning or breeding pond, incubation hatching pond, and hatching receiving pond. This system is designed for fish breeding and incubation. Now, how does it work? After pituitary injection, the male and female brooders are released into the spawning pool. The hatchery is made ready prior to the introduction of eggs, and the level of water is maintained by adjusting the outlet pipe. The eggs may be brought mechanically from the spawning pool or may be drained directly into the outer chamber of the hatchery through underground pipe systems. The eggs in the outer chamber are kept in circulating condition due to the circulation of water made possible by the circulatory arrangement of pipes fitted on the floor. Continuous water circulation in the incubation tank helps to maintain a high oxygen level of the water and churning of eggs throughout. The chosen flow rate of water helps to maintain the required temperature necessary for high hatching success. The flow rate was increased during hatching because of high oxygen demand during the process. At about 11 to 12 hours after fertilization, hatching of the embryo starts and gets completed within 4 hours. After all the eggs have hatched, the flow rate of water was decreased. To avoid bacterial disintegration, the eggshells, unfertilized eggs, dead hatchlings, etc. are removed periodically from the incubation chamber. This is done either by siphoning or by dipping a jute rope with a weight into the outer chamber. The circulation eggshells get attached to the jute rope, which is periodically taken out. The eggshells removed and the rope is again put into the outer chamber till nearly all the eggshells are removed. Some unfertilized eggs remain behind in the hatcheries, and these are the focus for fungal infection caused by saprolegnia. Saprolegnia. A common method of controlling fungal disease in the incubator is by the application of malachite green at a concentration of 0.02 gram per liter of water for about 20 to 25 minutes after stopping the flow of water. When the flow is resumed, the chemical is washed out from the incubator. Now, what are the merits of this? Chinese hatchery ensures a high hatching success, that is 98% as good and clean water replacement is possible. Water flow and thus the temperature can be maintained during hatching. The Chinese hatchery can be directly connected to the breeding pool on one hand and to the nursery pond on the other. Therefore, it ensures less damage of fertilized eggs. And spawns at the time of transfer. It minimizes the labor cost. Much larger quantity of eggs can be accommodated. Now, what are the demerits? Due to the absence of proper spawnery, total removal of shells, unhatched eggs, and dead spawns and larvae are not possible. Initial cost of the setup is very high. Proper technical application has to be maintained regarding fitting of pipes, redirection, and amount of water flow. Next is D variety hatcheries. The seed production is dependent on nature, but the problem has now been solved with the evolving of modern hatchery model CIFE D81. It is now possible to breed fish without rains in this modern hatchery. Thus, we have become independent of the monsoons and natural environment. The brooders are kept in the breeding unit, while hatching is done in jars having control over silt, oxygen, temperature, and metabolites. This hatchery system consists of breeding and hatchery units. After 4 hours of spawning, the eggs are transferred to the egg vessel, which is fixed in the hatchery jar. Then about 2 to 2.5 lakh eggs can be accommodated in each hatchery jar, depending on the species. Continuous mild aeration and water flow are maintained in the jars for free floating of eggs. The rate of water flow is maintained at 1 to 2 liter per minute. The eggs hatch within 14 hours. When the hatching is complete, the egg container with the shells is removed. Then the flow of rate of water in the jars is slightly increased for speedy transfer of the hatchlings into the spawn receiving tank. The remaining hatchlings, if any, are transferred into the hopper by siphoning with a 25 mm diameter pipe. Once the jar is emptied, water flow in the hatchery jars is stopped. The spray is arranged around the upper edge of the spawn receiving tank and is kept in operation to ensure high level of dissolved oxygen and low temperature. Coming to aeration, the aeration and spray are kept in operation continuously until the yolk sacs of the hatchings are absorbed, which normally takes 2 days. The percentage of hatching is 93 to 98 percentage. Now, what are the disadvantages? Material used is low density polyethylene, hence difficult to break, easy to pack and transport to different interior places. Controlled temperature system is introduced. Metabolites are removed from the circulating water by filtration. Due to the additional aeration, oxygen in water is raised to 7 to 9 ppm. Even when fertilization of eggs is low, the hatching rate is high. The system ensures breeding and hatching without rains and monsoon. Due to the filtration, the water is free from sediments and silt. Each jar has a provision for independent regulation of aeration and water flow. In case of mortality, pollution or disease in any of the jars, it can be isolated from the rest of the system. The common carp eggs normally hatch in 72 hours, but in this system, these hatch out within 42 hours. So these are the advantages. Now, transportation of breeders and seeds. Transportation of breeders, fry and fingerlings is a common phenomenon in fish culture. The fish seed are transported from hatchery units to the fish farm to rear them in culture systems. Now, the causes of fish mortality during transportation. Effect of carbon dioxide and dissolved oxygen. Mortality of fish seed may be expected during transportation. It is mainly due to the depletion of dissolved oxygen and accumulation of gases like ammonia and carbon dioxide in the medium of fish seed carriers. These gases are lethal as they may reduce the oxygen carrying capacity of fish blood. However, the lethal limits owing to carbon dioxide in fish depend on the level of dissolved oxygen. It has been reported that fry of more than 40 mm in size may die at 15 ppm of carbon dioxide at a dissolved oxygen level of less than 1 ppm. Such fry may die only at 200 ppm. If the dissolved oxygen is around 2 ppm, carbon dioxide given out during respiration dissolves in water and renders it more and more acidic, which is injurious to fish. In transport of the fish, the storage of oxygen has to be tackled either by replenishing the oxygen which is used up or by economizing its use by regulating the number of fish seed and by reducing its oxygen demand. Now, what is the effect of ammonia? The large amount of NH3 is excreted by fishes if ammonia concentration is 220 ppm. Total mortality of fish occurs in oxygen-packed packets. As NH3 increases in water, the oxygen content of blood decreases and CO2 content increases. NH3 interferes with oxygen carbon dioxide exchange capacity of blood with the outside medium. The rate of NH3 excretion increases 10 times with a rise in water temperature from 8 to 15 degrees centigrade. Increase in water temperature and decrease of dissolved oxygen reduce the tolerance of fish to NH3. Then effect of temperature. Temperature has a distinct effect on oxygen utilized by the fish. Metabolism increases continuously with increased temperature till the attainment of lethal temperature limit. Each species displays its own characteristic rate of increase at a given range of temperature. Fish, prawn, and their seed face hyperactivity during transportation. As a result, lactic acid tends to accumulate in their tissues and severe oxygen deaths are created. Fish take a long time to overcome this oxygen debt even in their natural life in ponds and other habitats. This may be due to the death of fish after few hours after handling, transport, and liberation, even in oxygen-rich water. Hence, the use of sedatives is most important in modern live fish transport technology. Next, techniques of transport. Several types of containers are used in the transport of fish seed. These are mud pots, round tin carriers, double tin carriers, oxygen tin carriers, and tanks fitted on lorries. The containers are transported on bicycles, carts, rickshaws, boats, lorries, trains, and aeroplanes. Then, mud pots. Mud pots are commonly used in Assam, West Bengal, and Orissa for transporting spawn, fry, and fingerlings. This is a traditional method. Mud pots of about 15 liters capacity are used for transportation of fish seed. The pots are filled with water of spawning, ground to about two-thirds of their capacity. After filling the pot with water, about 50,000 spawn are introduced. It is better to condition the spawn in the hapas for about 3 days without feeding prior to the transportation. Otherwise, due to feeding, more excreta is formed which pollutes the water in the pot, leading to the death of fish seed. To avoid the mortality of fish seed due to asphyxiation, water is changed once in every 5 hours. Round tin carriers. Round tin carriers are used for transport of fish seed from several years. The tin is made up of galvanized iron sheet. It is a round container having a diameter of 18 into 8 inches height. The lid has a number of small holes which are useful to get oxygen. This container has a capacity of 9 gallons of water, but is filled only 8 gallons of water. The seed is introduced into it and transported to various places. Double tin carriers. Double tin carriers are made up of galvanized iron and have two parts, outer and inner tins. The outer tin is open and with a handle. The inner tin is closed with a lid and entire tin has small openings. The inner tin is filled with water after keeping it in the outer tin. Then fish seed is introduced into it. It holds about 6 gallons of water and is generally used for carrying a small number of fish seed by hand. Then coming to tanks fitted on lorries. For road transport, lorries with one or two large tanks of suitable dimensions fitted at the rear can be advantageously used. This will facilitate seed transport problem to a large extent. Then we also have uses of anesthetics in transportation. So recent investigations have shown that the fish seed could be anesthetized for transportation for ensuring better survival rate. The purpose of this is to ensure that the fish seed survives for a longer period of time and also to minimize the concentration of toxic gases like ammonia and carbon dioxide in the medium by lowering the metabolic rate of the fish seed. Anesthetized fish seed have been found to survive for double the time of than fish not anesthetized seed, besides ensuring a better survival rate, which is about 90%. Carbonic acid has been found to be the best anesthetic compared to others such as quinaldine, sodium amytal, urethane, veronical chlorobutanol, and TMS 222. Carbonic acid is not only cheap but also safe and easy to use. To about 8 liters of water in bag containing fry, 8 ml of 7% sodium carbonate solution and 8 ml of 4% sulfuric acid are added to so as to produce 500 ppm concentration of carbonic acid. This anesthetized bag should be immediately filled with oxygen. Now, collection and transportation of brood shrimp. Live brood shrimp can be collected either from the commercial trawlers or from traditional boat operations. In collecting live brood shrimp, the trawling operation should not exceed 30 minutes. In practice, this may not be possible since no trawler operator will be willing to do this. They collect whatever live brood shrimp specimens are available in a trawl hall. Hatchery owners would have to provide large fiberglass or syntex tanks with portable battery operated aerators to the trawlers to enable them to maintain the brood shrimp in a healthy and live condition. Non mechanized boats normally operate gill nets to collect shrimps. The nets are usually set at sunset and the catch is checked at midnight and at sunrise. The traditional fishermen collect brood shrimp in live condition by using their own indigenous methods. Usually, the brood shrimp are packed and transported by the following methods. Live animal holding tanks in trucks with provision for continuous aeration are used. Different types of tanks also can be used for this purpose. Brood shrimp are also held in double-walled plastic bags with cool seawater, that is 22 to 24 degrees centigrade, and oxygen and packed in styrofoam boxes. The rostrum of shrimps is covered with small rubber tube to prevent puncturing of plastic bags, or the spawners are immobilized in separate PVC bags. With this, we have come to the end of the chapter. These are the model examination questions frequently we can see. Write in detail about carp hatchery. Write about earthen hatching pits. Write about earthen pot hatching. Write in detail about glass jar hatcheries. Write about working principle of glass jar hatcheries. Write about hatching hapas. Write about vertical jar hatcheries. Write about Chinese hatchery. Write in detail about working principle of Chinese hatchery. Write about D variety hatchery. Write about portable FRP carp hatchery setup. Write in detail the causes of mortality during transport. Write about techniques to be followed in transport. And write about transport of breeders. Thank you.